rustle too much as a result. So if we talk about brain imaging, so ways of imaging the brain during life, what options do we have? Well, when most people think about brain imaging, they'll think about imaging the structure of the brain, what the brain looks like. And there are two main ways we use that, which is CT scanning and MRI, which everyone will have heard of. There are some other emerging ways as well, or some of them are quite established, which is about looking how the brain is functioning. And that's either how the brain is using glucose to metabolize, but increasingly, actually, the abnormalities within the brain that might be specific to some of these diseases. And I'll come along to talk about some of those in due course. So why should we undertake structural brain imaging at all? Well, as many of you will know, in this country, we're looked over by NICE, the National Institute for Clinical Excellence, and NICE came out with some guidelines to help us with this and say that structural imaging should be used to assist in the diagnosis of dementia, to aid in the differentiations of the different types of dementia, and to exclude other cerebral pathology. So that's the guidance we're working to, and I'll go ahead and explain what we mean by that in a little bit more detail. And they go on to say that MRI scanning is the modality of choice, but we can use CT scanning as well. So where possible, we should use MRI scanning. And the reasons for doing that is that it gives us much more information, and it doesn't have any X-rays, which CT scanning does. Although there are some problems. Some people can't have MRI scanning if you've got pacemakers. It's a little bit noisy and a little bit claustrophobic for some people. So if we start from the bottom up, at the bottom of the first paragraph, excluding other cerebral pathology. So the brain is clearly a very, very complex organ, and the sorts of changes that we see in FTD, so changes in behavior and changes in speech, can occur for reasons other than a progressive dementia. And so this is a CT brain scan, and you don't need to be a neuroradiologist, I think, here to see this big tumour here, which is sitting in the frontal lobes of the brain. And if you have a tumour like that, that is going to produce quite a lot of behavioural change. But we wouldn't label that person as having FTD, and it may well be that you could operate and take that tumour out. To what extent it will improve matters is open to question. And here's another large tumour here, pressing on the front of the brain, which potentially can be removed. And also, if you've got bleeding around the brain, so a slow bleeding from the veins being ripped here, you can see this blood, the black areas around the brain is compressing the brain, and that can produce a wide variety of different symptoms. And so it's quite important, before we diagnose someone with FTD, a progressive degenerative process, that we make sure that these pathologies aren't there either. So if we are moving beyond just using imaging to exclude things that might mimic FTD, how can we go on and use brain scanning to help us make a diagnosis, and most importantly, to start distinguishing out the different forms of FTD and the different underlying pathologies that can cause this? And I know that Camilla's given you an overview, and you'll hear a little bit more about that, so apologies if there's a little repetition here. But I thought it might be helpful for us just to consider a little bit about what we mean by a degenerative condition, a degenerative dementia. We talk about neurodegeneration. What do we mean by that? Well, a neurodegenerative condition are conditions which show similar characteristics. So they start in a pre-symptomatic phase with proteins that are often normal proteins within the brain, starting to misfold and to accumulate. This in turn, over a period usually of many years, causes the brain cells, the neurons, to break down in various, sorry, my misspelling, networks here within the brain. I spotted it a bit too late. And that leads in turn to brain shrinkage. And this whole process, in most of the neurodegenerative conditions, takes many, many years. And if we are being purists about this, to make a diagnosis of any specific neurodegenerative condition, of which there are many, what we're trying ultimately to do is to predict what we'll see under the microscope. And so these are some of the proteins that we'll see in the different neurodegenerative diseases. So the commonest neurodegenerative disease is Alzheimer's. And we see these amyloid plaques and accumulations of a protein called tau there.
John,
Yeah.
what does misfold actually mean?
So the way that proteins are formed is proteins are a string of amino acids. And that string of amino acids folds up into a very, very specific structure. And that structure, which is often a physical structure, will determine how those proteins interact with each other, with enzymes and so forth. And so when proteins start misfolding, they tend to clump together. And it's the clumping together of those proteins, either outside or within the cells, that cause the cells at the neurons to start dying and to. But actually, it's a very good question, because exactly why that happens isn't entirely clear, and a lot of basic research is going on to why. But it's a very, very good question. So what we're often trying to do is to work out what these misfolded proteins are. And we know what they are in Alzheimer's disease, which is the commonest. And here is a Lewy body, which is we see in dementia with Lewy body. And then the problem with the sorts of diseases that everyone's dealing with here is that there isn't just one pathology. Sometimes it can be related to Alzheimer's pathology. Sometimes it can be related to tau pathology, of which there are several different types. And I think Dr. Aurora will speak to you a bit more about that later. And then we've also got this other protein that's been determined relatively recently called TDP-43, which comes in lots of different forms. So we've got lots and lots of different proteins that can fold up and cause this process to start. But what's important is that this isn't a completely random process. And whilst there's not a perfect one-to-one match, generally speaking, the proteins that fold up tend to fold up in specific parts of the brain and cause the sorts of symptoms that we see. And that leads to the brain shrinkage that we see on the scans, and that in itself is helpful when we try to use the brain scanning to make specific diagnoses. We'll come to that. Thank you. So if we look at structural imaging, so this is an MRI scan, so we're looking at the end of that process, not at the proteins misfolding, not at the networks, we're looking at the atrophy, the brain shrinkage. And this is a scan from a patient with Alzheimer's disease. And what we're doing here is we're slicing somebody through the middle of the brain here. So we'd find the ears out here, and this is the top, this is the left, and that's the right. And this black area is the waterway system, the ventricle system within the brain. And here, folded up here, are the hippocampi, which are the structures that are involved in day-to-day memory, finding your way around. And those are the neuronal structures that are typically involved in patients with Alzheimer's disease, which explains why patients with Alzheimer's disease often will present with problems with their day-to-day memory or finding their way around. And actually, the presence of shrinkage of these structures here has got utility in diagnosing Alzheimer's disease or diagnosing it more accurately and earlier. As we said with FTD or FTLD, frontotemporal lobar degeneration, as everybody I'm sure here will be aware and will have heard already, is that this is an extremely complex group of different disorders which are bundled together. And you can bundle them together in several different ways. You can bundle them together on what the symptoms look like. And we think about behavioral variant frontotemporal dementia, and of course within that spectrum there are people who are more apathetic and more disinhibited. Then we talk about the speech variants, people who have fluent but rather empty speech because they've lost word meaning, those who have very effortful speech, and the people who have word-finding pauses and naming difficulties. So this is clearly a complex classification based on the clinical features. It's equally complex, as I've said, because we've got different proteins under the microscope. We've got tau, two different types, TDP-43 of several different types, and Alzheimer pathology, which can cause these different symptoms as well. And furthermore, as people here will know, and again, John will talk about a little bit more detail later on, about 1 in 3 people have got a good family history of frontotemporal dementia, particularly the behavioural variant that runs in the families. And of those who have familial FTD, we now know that there are 3 genes. One, mutations in the tau gene, one in progranulin, and one in this third and fairly recently discovered gene, which is C9orf72. So you can see that as when we see somebody with this spectrum of disorders, there's lots of things we're trying to work out in our heads. Is there a problem? What's the underlying pathology, which may be useful and will become particularly useful when we have specific trials which may target these different pathologies? What is the group of symptoms that the individual has? Because that's the thing that's important to them, so that we can guide and provide prognosis and provide information, and making sure that we're not missing something that we could be treating like a tumour. So those are all things that we are trying to assess when we are using scanning. So just by way of some examples about some of the MRI scans, trying to tease these various aspects apart. This is a scan from a patient with frontotemporal dementia, and this one happens to be somebody who had tau pathology. So we're now looking right at the front of the brain. You can see here are the eyes, and these are the front of the brain. And you can see that there are great gaps here, this black area around the brain, which is full of spinal fluid, because the frontal lobes of the brain have shrunken down. And it's the frontal lobes of the brain, hence the name, which are particularly involved in social interactions and behavior, and this gives rise, or this is the end product of those protein accumulation and network breaking down, and correlates well with the symptoms. And this is another patient who's also got behavioural variant, but has got, due to a different protein abnormality, which is called TDP43. You can see this is a slightly different pattern of shrinkage. So here's the left side and here's the right side, and you can see this stage there's a bit of asymmetry. So if you look at this temporal lobe here, it's a little shrunken down compared to the other one, and it's a little bit shrunken here compared to here. So we've got some asymmetry. This is also presenting with behavioural features. And this is a patient again presenting with behavioural features, who this time has got very profound shrinkage on the right-hand side of the brain here. So you can see here's the left side, and if you compare across, you can see this ventricle is very expanded, this temporal lobe is much more shrunken than this one, so a lot of asymmetry. And so when we are trying to determine whether a patient has the FTD spectrum, individuals who have lots of atrophy at the front of the brain compared to the back of the brain or in the memory areas of the brain are more likely to have FTD than, say, Alzheimer's disease or dementia with Lewy bodies. And patients who have a lot of asymmetry, one side of the brain affected more than the other, are more likely to have disorders in this spectrum. The reasons for this are unknown and are of great interest to try and help us understand why these changes occur and why it leads to the different symptoms. What about some of the genetic causes? Well, sometimes these can give rather unusual patterns. This is somebody who is presenting with a tau mutation, who actually presented with a lot of memory problems, and you can see why, because I've told you that these are the memory areas of the brain, and they're very, very shrunken down. Actually, rather more shrunken than we see in Alzheimer's disease. So the situation gets yet more complicated, because sometimes the scans can look a bit like Alzheimer's disease, although there are some subtle changes. This is somebody who's got a progranulin mutation, so another mutation cause, and you can see again this profound asymmetry. So this would give make us think about frontotemporal dementia, because there's this asymmetry with the right side of the brain being affected much more than the left. So those are sort of thinking mainly about the behavioural forms. What about the speech variants, so people who have the different speech forms? So patients who have very effortful speech, can't get their words out, often have surprisingly normal-looking scans. So a little bit of asymmetry, a little bit of shrinkage around this area compared to this, remembering that the left side of the brain controls speech, but often fairly unremarkable. Whereas patients with semantic dementia, who often have very fluent speech but difficulties with naming, often have very shrunken scans, mainly on the left than the right, this temporal lobe here being very, very, very thinned, despite actually often functioning at a reasonably at a high level. So we have a mismatch between what the scans look like and how individual patients are. But the scan appearances can be very helpful when we're trying to put the pieces together. And patients who have the effortful, slow speech with pauses, again, the scans can be quite abnormal. There's often a bit of asymmetry, with the left side of the brain affected more than the right, but doesn't look too bad at all. So the scans can give us very, very important clues, these structural scans, but often they won't give us the whole picture. And again, none of these scans is absolutely diagnostic or can allow us to absolutely predict the underlying pathology. So it's taken together with the information that we get when we examine patients and take a history. So that's about structural scanning. So what other sorts of scanning are now available? Well, I talked to you a little bit about functional scans. So these scans, and here is an FDG, which is a glucose PET scan. So this is a different type of scanning altogether. So this is when we inject a small amount of tracer, which has got a label that marks glucose, and we inject it in, and we wait for it to cross into the brain, and then we take a picture about how the brain is actually using glucose. And here's an example of an FDG PET scan. And the brain around the outside should be red all over. But you can see here that there's a lot of areas here. And in fact, this is actually a patient with an atypical form of Alzheimer's disease who has lost, not metabolizing at all on that side of the brain or at the back of the brain here. So that can show us which bits of the brain are functioning well and which bits aren't. And in answer to the earlier question about whether we can only see some of these proteins at post-mortem, very exciting advances over the last few years, allowing us to get actually to see some of the underlying molecular pathology, the changes that we could only previously see under a microscope using PET scanning. And the one that's come furthest is for scanning for amyloid. So this is an amyloid PET scan. So amyloid is the principal, it's probably one of the two prime and probably the principal protein abnormality in Alzheimer's disease. So here's somebody who is healthy and normal. There's a little bit of uptake deep in the brain. And here's someone with Alzheimer's disease. And you can see that we are imaging here this abnormal folded protein during life. So this is now licensed for clinical use. It's extremely difficult to get hold of because it's extremely expensive. But in terms of ruling somebody in and out of having Alzheimer's disease pathology, if someone's got dementia, this can be extremely helpful. It needs to be a bit careful because there are probably some elderly individuals around, a significant proportion, who may well have these changes, who may in due course actually be on their way to getting Alzheimer's disease, who don't have any symptoms. But in the context of somebody with a dementia or with cognitive impairment, if the scan looks like this, then it's very, very strong evidence that there is underlying Alzheimer's disease. And conversely, if it's not, then it pushes you away from Alzheimer's disease and makes you think more about FTD and the other neurodegenerative disorders. So I'd now like just to finish up with showing you some early results of a study that you here in this room have partially funded, along with support from one of our industrial collaborators as well, and we're extremely grateful for this. And this was a study that we set up several years ago and actually turned out to be extremely difficult to do, because at the time we were doing this, we were using this amyloid tracer which wasn't licensed for clinical use, which meant we had to do clinical trials and all sorts. So it's taken rather a long time to do. But the study was really to start trying to do some comparisons about all of these different ways of imaging the brain in different degenerative diseases. And we involved 25 individuals. There were 5 who were controls. We used 5 patients with a form of Alzheimer's disease, really so that we could be sure that the tracer was working well. And then we tried to concentrate on the group of individuals when often the diagnosis is most difficult and understanding what the underlying pathology is most difficult. So those are people who have speech problems. So we had representatives of the major speech groups at that stage. And to give you some examples of what we see with the different scanning modalities. So here is one of our healthy controls. And at the top, I'm showing you what the amyloid scan looks like. And then underneath, what the FDGs, so the glucose uptake. And here is the structural brain scan. And the individuals who took part in this came for a scan on 3 consecutive days. So they had an MRI scan on one day, an amyloid scan the next, and a glucose scan the third day. So very involved. In fact, we did lots and lots of different types of MRI, which I won't go into at this stage. But you can see here that there's no amyloid in the brain here, that the brain is metabolizing very well, and the brain structure looks very normal. And here's somebody who's got semantic dementia. There's no amyloid in the brain here, because semantic dementia is usually associated with a different protein, TDP43. You can see here that there's no metabolism here in that left side of the brain here, and there's a lot of shrinkage on the scan in that area as well. But in fact, that the area where there's not so much glucose uptake exceeds where there's been brain shrinkage. So this is potentially brain that's vulnerable but not yet completely disappeared. This is someone with a progressive aphasia, no amyloid in the brain. Again, quite a widespread pattern of impaired metabolism and a little shrinkage on the left-hand side of the brain there. Whereas all the semantic dementia individuals who took part didn't have amyloid in the brain, some of those who had aphasia did have amyloid in the brain, making it clear that with virtually identical scans and clinical syndromes, it's difficult to predict who's got amyloid in the brain and who hasn't. So here is somebody who has got lots of amyloid in the brain, who's got a similar pattern of metabolism and a similar pattern of shrinkage. Here is somebody who's got the hesitant speech form of FTD, which is usually associated with Alzheimer's disease. And here you can see lots of amyloid in the brain. Metabolism changes, we look fairly similar to those two, and a scan that's not so dissimilar. And here, by comparison, is somebody with the posterior variant of Alzheimer's disease, lots of amyloid in the brain, lots of metabolism loss, particularly at the back, but extending forward, and some shrinkage at the back as well. So you can see here in these examples that each of the different imaging modalities is giving you different information. Some about the underlying protein, in this case the Alzheimer's, some about the pattern of metabolism, and some about the areas of brain shrinkage. And if we try and put these together, so these are small groups, but if we try and map the areas that are changing in the individuals, we can see, and here this is a representation on a brain here, that the patients with semantic dementia have got huge atrophy here around the left temporal lobe. And those with the progressive aphasia, it's more patchy, extends over the left-hand side, but a little bit further back. And individuals with the logopenic, the slow form of he said with speech impairment. The speech impairment and naming problems in Alzheimer's disease, it's similar but extending a little further back. What about the glucose metabolism? Well, very focal areas in the brain around the left side of the brain here where the brain is not using glucose so well. In the logopenic cases, much further back here in a pattern that looks much more like we'd expect in typical Alzheimer's disease. And the progressive aphasic cases is sort of somewhere in between. And finally, the amyloid deposition. No amyloid in any of the semantic dementia cases. Lots of amyloid in the logopenic aphasic cases, and just a speck of positivity because there was 1 out of the 5 patients here with the progressive aphasia who had amyloid, so much more of a mixed group. So I hope I've given you an overview here about why we undertake imaging in FTD and why it might be helpful, the different modalities that we have open to us. That these different imaging modalities don't do the same thing. They provide complementary information about structure, function, and the underlying proteins. And just to say that this is really just the beginning. So I've said before that tau is one of the other major proteins, both in Alzheimer's disease, but also in FTD, and we now have tracers that can label tau, and work that's going on in Cambridge, and we'll be starting here shortly, is going to be using these new PET tracers to look at tau, and it's likely that we're going to have PET tracers for Lewy bodies and TDP43 and the other pathologies soon, allowing us to get a really good handle on all of the different processes that lead to the development of these conditions. And as well as that, I haven't touched on is the other MRI techniques, which allow us to look at a vast range of different aspects of tissue structure, the connections between the brains, which is all ongoing work. So I'll finish there. I'd like to thank everybody who's taken part in the research. This was, a lot of work for people to come for 3 days, and very, very grateful. All of my colleagues. I'd like to thank everybody here, and particularly Lynn and Duncan for raising money towards this, and our colleagues at Eli Lilly. And I'm very happy to take any questions. Thank you.